So, a very warm welcome to our friends and colleagues from over 15 countries across the world. Very warm welcome to our latest NSF webinar, which is entitled The Hot Topics in Good Manufacturing Practices for Biotech and Biological Processing. So, a very warm welcome, everybody. Bonjour, guten tag, hola, namaste, and gamma jobat. I've got quite a lot to get through over the next 30 minutes or so, so this is going to be a pretty fast whistle-stop tour of these top 10 hot topics. So buckle up, enjoy the ride, and let's see what you think. So you know who we are. We are NSF, Pharma Services, and we are a leading GMP consultancy across these three main work streams. Specialising really in transformational change. You also know me, of course. I'm John Johnson. I'm Vice President of NSF Health Sciences, and I'm going to be bringing to you quite an interesting webinar on biotech processing today, and hopefully this is going to be of use to you. So, we're going to cover these top 10 concerns. These concerns when we're looking to interpret, define, and execute GMP across what we would like to define as a typical biotech process, but actually there's no such thing as typical. There is so much variation in the way that biotech products are manufactured, and that's a key message here, that we will try and generalize, we'll try and give you some good generic overview key messages, but you have to think hard for yourself, think hard about your own process and your own facility and your own particular product attributes, and utilizing, of course, as I always say, absolutely world-class risk identification, risk evaluation, and risk management. And you might well remember my last webinar on quality risk management, where I said there was absolutely no substitute for being active in seeking out risk, calling out the issues. Remember, I often talk about how an issue that is well defined is an issue that is half resolved. So taking the time to really identify the problem, call it out, and it always drives you to take the right preventive action for the long term. So all the way through this webinar, we're going to be talking about risk management across your biotech process. And if you want more information about this, you want more of a deep dive, we've got a 4-day course that we're going to be running in Nibet in Dublin in September, and I've provided you there the link to that course where we go through this in obviously a lot more detail than we can in 30 minutes. So I've mentioned before it's hard to define a typical process, but there are some generic elements of a biotech process that are common. And you can see these across this stylized large molecule shown on the graphic on slide 5. And the main thing I wanted to mention here, first of all, is that there is absolutely no substitute at all for effective process design, effective validation, and real-time monitoring. That's absolutely crucial in your biotech process. Equally, there's no excuses for not knowing what the CGMP expectations are, not appreciating how to interpret them, or for not really understanding the product quality attributes that you need to assure during the validation and execution of the manufacturing and testing process. And of course, time and again, what really differentiates our success is the level of risk management that we put to our process. So, moving on to slide 6, obviously most biotech molecules are intended in their final formulation to be a sterile product, and it's always important, I think, when you're thinking of your biotech drug substance, to think about the sterile drug product key quality attributes. And of course, these are obvious, but often not really considered in full, and not really assessed in terms of batch document definition or process validation definition. And some of these key attributes are obvious, but some are less obvious. And it's important to have a proper scientifically justifiable list of the things that you're looking for in your final drug product, so that you can determine what your final drug substance must actually achieve. So let's go through these top 10 subjects. The first hot topic really is that you've got to be able to prove that the product you're making now is bioequivalent to the batches that you made for clinical trials many years ago. And what I mean by that is you cannot actually make an identical structure, or you can't actually prove that structure is identical because of its complexity. The complexity of the primary, secondary, and tertiary structure of a biomolecule makes it very, very difficult to prove bioidentical. You can only really prove bioequivalence. So how do you go about that? Well, if there was a single tattoo that I would recommend for all of us in bio and pharma biotech manufacturing, it would be the reference to the FDA quality system inspection technique approach, which is 7356.002. So if you don't have that tattooed on your forearm and look at it every day and reference that key document on the internet under fda.gov, then you're really missing a trick, because that document 7356.002 describes these 6 systems that are inspected each and every time by the FDA during a pre-approval inspection, and a selection of these will be inspected each and every time during a general GMP inspection. So what are they actually trying to show? The first thing they're trying to show is that you've got clear links between what you do today, how you manufacture your batches today, to how you manufactured your batches during process validation, and how you manufactured your batches during the pivotal clinical trial manufacturing program. And of course, all of that involves a look back. You're looking back at the past. So you're looking at records, qualification and validation records, process validation records, batch records, QC records, to confirm that what you're doing today is equivalent to and has effective change control to what was done in the past. Equally, of course, by looking at policies, procedures, instructions, the inspector can also see how you would manufacture future batches. So again, making that link to what you're doing today to what you'll do in the future. So making sure that you've got very clear, scientifically justifiable links from what you did during pivotal batches, during validation batches, during today's batches, and also into the future. And fundamentally, during that inspection, they'll want to confirm and verify license compliance, i.e., are you doing what was actually submitted against CGMP expectations, and fundamentally to show or at least verify proof that you have dealt with any perceived risk that may involve either misbranding issues or adulteration issues. And I think that single slide is so useful in preparing for any inspection, and also preparing yourself to prove that your particular batch or your particular campaign of batches is bioequivalent to previous batches. So, item number 2, has the firm actually undertaken adequate risk assessment and mitigated any risks that avoid misbranding? So what you're trying to do here is trying to prove that the product is what you intended it to be, and it is meeting all of its specifications, labeling requirements, and is made according to the submission. So in effect, you're making sure that you're claiming something you can prove is actually in place. So what do we mean by misbranding? Well, it may be that the product is the wrong strength, or it's mislabeled, or it didn't meet GMP, or in some ways not accurately described as per the submission. And the way I often describe this is if you were a vegetarian and you were ordering a vegetarian meal in a restaurant, and being vegetarian was important to you for religious or personal or dietary reasons, you'd be really unhappy if your vegetarian curry was cooked using animal fats. And it's exactly the same with pharma biotech. If you're manufacturing a complex biomolecule, you have to be able to prove it is exactly what you set out to make in the first place. Third key item, of course, is all around adulteration, avoiding the main forms of adulteration during the full supply chain, really from the originating biomolecule through to the purified drug substance. And what I show on slide 12 here is that the onus of proof is on us as the manufacturer. We have to be able to prove that the product is not contaminated. It's not up to an inspector, an auditor, a regulatory inspector to prove that your product is contaminated. It's our job to prove that it is not. So, of course, when you think about contamination, you're thinking about non-viable particles and all of the sources that may form that particular risk, and of course viable particles. And many firms now use some really good tools, some best practice to make sure that these risks are well covered. So for example, with non-viable particles, many companies now make a very detailed inventory of the product contact surfaces. They list the materials of contact of every product contact surface, and they do a risk assessment of any potential sources of contamination from each and every material of construction, wherever that may be. Similarly, for viable particles, and as you've seen in previous webinars from me, the best practice by far is to utilize a contamination control strategy. Just the same way as I explained when I described the requirements of a contamination control strategy as part of EU GMP Volume 4, Annex 1. So by utilizing these two proactive risk assessment techniques, you can be sure that you've eliminated the main sources of contamination within your process. Item number 4, well, the key FDA quote from CBER way back in the 1980s, which I think was one of the most instrumental quotes I've ever seen in this field, was very, very simple. And the quote was, "The product is the process. The product is the process." So, if your process has changed, can you actually prove that the product hasn't changed too? We all know that these biomolecules are very fragile, they're very complex, they are very susceptible to change. And if a change occurs within your process, how do you know for sure that your product is unchanged? It's still going to give you the quality attributes that you expect to see. So your change management process, how you confirm that each and every key step in your very complex multiple chain of bio processes, how do you confirm that each of those parts of the chain have been executed flawlessly, right first time, to validated and registered settings? And of course, Donald Rumsfeld helps us a little bit here with change control. And I think it's a nice message on this slide that shows how if you develop your products effectively, if you validate your products effectively, if you gain the product knowledge across the process train effectively, change control becomes far, far easier. So if you look at this, if you look at, for example, the early stages of development down here in the graph. So you don't have the product knowledge, the product history. It could be a new biological entity for you. So there's going to be a lot of unknowns. There are a lot of things we don't know. So in terms of change control, it's very difficult to see what changes are significant and critical and what changes may not be. As you move up the product life cycle, you get to a stage where you have more known unknowns. So there are things that we know, we don't know. And that's when you're moving into more advanced development, leading into process validation. So the level of ambiguity in the knowledge of your process is reducing, your knowledge is growing, so change control therefore becomes a lot easier. And of course, by the time you're up at the top of this chart and you've completed your process validation and you have a licensed product, you will have a lot of known knowns, and you'll be able to assess change much, much easier than you will when you're down at this development level. So the key message here is that regardless of where you are in the product life cycle, change control is absolutely crucial, and will be a hot topic at any future inspection. Number 5, of course, issues occur. Life is real, and in real life, unexpected, unpredicted events occur, and these types of deviations have got to be properly investigated. And the investigation of those unexpected events is absolutely critical. And you've probably been in similar meetings to me where, with a lack of discipline, with a lack of knowledge, the people around the table almost seem to use a spin-a-course tool to jump to conclusions as to why an issue has occurred. Everyone's in a rush. Everyone wants to get to the solution as quickly as possible. And if you don't use some very disciplined, meticulous techniques, it's very easy to jump to conclusions and therefore drive the wrong kappa. So the 3 key requirements here to avoid this is obviously to use great root cause analysis techniques, to instigate and promote and to enforce a blame-free culture, and of course to set up your quality system and your way of working as an environment for institutional learning, so that you don't use some of these knee-jerk reasons for classifying a deviation, identifying the root cause, and then identifying kappa. It's always important to think about the real root causes, noting that there is never a single root cause, always a multiple one. Moving on to number 6, of course, your facility is going to be really important in your biotech process. Is it well designed? Is it spacious? Is the process and product flow effective? And has it been validated and monitored to minimize any adulteration risks? So when you look at your facility concerns across either design or monitoring, it's pretty obvious that you can see all of this via your regular Gemba walks, by your internal audit, or by third-party GMP inspections. And you can see for yourself whether these things have been taken care of properly. A key message here specific to biotech is that real-time supervision and a QA presence on the shop floor who can react to issues quickly, methodically, utilizing the subject matter experts at shop floor level is absolutely critical. And a more hands-on collaborative QA approach always pays dividends in this particular environment. So number 7, what about our QC concerns? Our QC concerns being, well, whenever you take a sample, have you proven that the process of taking the sample has not changed the product itself? Is that sampling method validated? Is the assay and instructions associated with the assay well developed, error-proofed, validated and properly trained out? And particularly for the complex bioassays that we often see in this particular area of our industry, how have you gone about checking the proficiency of the operators who are going to, or analysts that are going to execute that particular analytical method? So when you think about your QC concerns, what would they be? Well, the first one would be, when you look at a particular bioassay or a biomethod, can that assay actually detect the product attributes that you're looking for? Can it detect evidence of misbranding or adulteration of the various forms? And is it going to be an insightful method for any particular process change, deviation, or impurity that you're looking for? Of course, when you think about the method itself, thinking about how complex they are, have you got a method that's been designed there that recognizes and also eliminates sources of error? Many of the bioassays are very dependent on the analytical expertise, so making sure you make that method as simple and as error-proofed as possible can be incredibly important. Needless to say, with other pharma biotech products as well, your systems for dealing with the investigation of out-of-specification results, dealing with how analysts are qualified and how they're checked for proficiency, not just on induction, but also as a repeated proficiency test is obviously very important indeed. And I think what's interesting as well is that many of the bioassay methods that we see out there are not straightforward in terms of validation, because ICHQ2 is a fantastic reference document for validating classical small molecule products where the characterization is relatively straightforward. And I'm thinking here in particular, for example, an HPLC method. But when you're looking at bioassay methods that are looking to elucidate primary, secondary, and tertiary structure, we know that there are many parts of ICHQ2 that just can't be followed. So how do you justify that? How do you look for other insightful ways of validating your bioassay? Moving on to hot topic number 8. We see, of course, out there in industry that many of the bioprocesses that we see have certain specialist parts that are outsourced. So these may be outsourced to a different time zone, either within your company or outside of your company with an approved partner. And in those cases, how on earth do you oversee the process? How do you know for sure the process is being performed exactly how you wanted it to be, how the instructions demanded, and how you expected it to be from the point of view of the process description? So if you're actually keeping a key step within your own manufacturing site, it's relatively easy for you to look at various forms of PLC or real-time monitoring and to be sure that your critical process parameters are being respected. But how do you do that when that process is being performed in a different time zone? Also, of course, by perhaps a different organization with a different culture, different pharmaceutical quality system, and different ways of working. So, of course, when that is the case, inspectors, license holders, qualified persons are going to be very interested in the way outsourcing was performed. So thinking about how the supplier was selected, qualified and monitored, what types of technical agreement requirements would be in place. Would you, for example, have a person in plant during key steps? How often would you do your GMP inspections or your short interval control audits? How often would you do gamble walks? Would you indeed have a full-time person in plant there at the facility? But again, in any case, it all comes down to communication, good culture, having an open management process and mindset, and making sure that you've got really effective risk identification and effective change management processes. So that you've got those clear links from your bio batch in the past, your validation batches in the past, and also your current and future manufacturing. So having a really close eye on outsourcing is absolutely crucial for biotech in many, many cases. Moving on to item number 9, of course, we've already said that bioproducts are often subject to very complex processes, and those processes need short interval control. So how do you make sure that your key staff are performing continuous professional development? How do you make sure they're refreshing their on-the-job skills and their knowledge so that they can detect issues and react to issues in the right way each and every time? And of course, when we think about this, we're thinking about the key staff, the subject matter experts. Are they experienced? Do they have the expertise? Are they new to the job? Are there perhaps issues of staff turnover? It can make a huge difference when your validation specialist or your process specialist is headhunted and joins an alternative competitor in this particular market. or is promoted into a role where they have no influence over the day-to-day quality of the process. So looking at staff turnover, looking at the demographics of the subject matter experts, very, very important. And of course, whenever new people are joining the organisation, how are they onboarded? How are they qualified to do their work? How are they given initial and ongoing proficiency checks or effectiveness checks? And how do we provide feedback to them so that they bring issues to you, they don't hide them, and that they continue to learn and you progress that person within a structure of effective knowledge management. And I mention here knowledge management on the bottom bullet here. You know, is this going to work for you, utilizing incredibly complex, wordy, heavyweight policies, procedures and instructions, or is there a more targeted way of working, almost like an internal Wikipedia or an internal, or an internal way of learning from the past? It's so important that you learn from past issues. So let's move on to number 10. And I think this is kind of interesting. When you look at your typical bioprocess and you look at the number of potential interactions with raw materials or product contact component materials, you can often find a huge amount of complexity. And we know that biomolecules are often very fragile. So if they interact or alter according to the quality of the raw materials, or a change to the product contact component material of construction, it could affect the quality of your product. It could either promote unwanted proteins within your protein mix, or alter the desired protein completely, and obviously affect the quality and efficacy of your final product. And the photograph here shows a single-use disposable buffer bag that is relatively straightforward in design, but believe it or not, has 6 different product contact materials of construction within it, each one of which has a specialist formulation, and each one of those 6 could have an effect on your biomolecule and its integrity. So of course, when you're thinking of your product contact materials or your raw materials, you've got to have a very effective supplier quality management system, a very effective incoming QC test regime, again, great change control, and being very aware of absorption and desorption from product contact materials. Of course, the other thing to consider as well is the raw materials, the buffers, the components, the tools, and how they can be completely segregated according to the viral status of your particular product. And many firms define their viral status as hot, medium, or cool within their process, and they will dedicate raw materials, components, staff, gowns, tools, equipment, so that there is no chance of cross-contamination across viral status areas. So very, very important to consider how your materials and components and consumables are used. In fact, probably 70% of the deviations that I personally was involved in about 10 years ago in an antibody drug conjugation facility were related to the control and quality of consumables. So, needless to say, we've got a lot to cover there in just 30 minutes, and the detail behind these 10 hot topics is going to be presented at our course on the 17th to the 20th of September, which is held at the world-class Biotech facility at NIBA in Dublin. And for 2019, that course has been completely revised and upgraded. You know, more than 22 of the 35 sessions have been revised or upgraded. We have 19 sessions of guidance, 2 fantastic guest speaker slots, 11 interactive team exercises where the delegates can learn from each other and gain insight from each other's expertise and experience in this key area. We have 2 really in-depth tours of the NIBUT facility where we challenge people to utilize and interpret what they've learned from the guidance sessions. And we have a new session on ATMPs as well. So it's going to be an exciting event. So what I'm going to leave you with is that bioprocessing is a complex train of process steps. It's actually impossible to rely just on QC testing as a determinant of quality. And overall, please remember that the product is the process. And if the process changes, the onus is on you to confirm that the product hasn't changed too. Absolutely critical message. So, you can gain access to us anytime you wish regarding our auditing services, our consultancy services, or our training services in this key field. And we'd love to see you at NIBUT in September this year, if you can come along. We, of course, always remind you of our public, internal, and customized training, our e-learning, and our auditing and consultancy work where we're so involved in transformational change. So I hope you've enjoyed this short webinar. I've tried to keep it to 30 minutes as I always do. Jim Morris will be doing exactly the same, at least in terms of keeping to 30 minutes when he does his webinar on the 23rd of July on the GMP standards of OTC drug manufacture. So I hope you can join Jim later on next month. Needless to say, thank you very much for listening and attending. We have plenty of other information on our resource library on the website, and I have a video about bioproduction also on that link. So thank you very much. I hope that was of interest. It was a whistle-stop tour. We covered 10 things in 30 minutes. Enjoy the rest of your day and hope to see you very soon. Take care. Bye-bye.
And bye.